Here we have a model of smooth muscle tissue. We actually have two models of smooth muscle. This is the larger of the two, and I'm just going to hold up right above it the other model. It's fairly small. There are three models that look similar to this, but I just wanted to be clear about the fact that this is our smooth muscle model. We're going to come back to that in just a second. So for our lab manual and for our lab exam, these are the features that you'll want to be familiar with. First of all, smooth muscle, which is characterized by spindle-shaped cell fibers. Something to note about muscle tissue is that cells are called fibers. So in this case, we'd call this a fiber. It's spindle-shaped or fusiform-shaped. It's got one nucleus right here, so it's uninucleated. And you'll notice on the outer segments of this model that there are no striations for this muscle. That's one of the key characteristics of smooth muscle. In our lab, the example of smooth muscle that we utilize comes from the small intestine, specifically the outer layers of the outer wall of the small intestine. And in the small intestine, there are two opposing layers of smooth muscle tissue. And I want to go ahead and show you what that would look like. This will help you when you're looking at your histology slides. Well, let me butcher my words. You're going to see a circular layer, which is going to be oriented in this fashion. So this circular layer, you're only going to see a cross-section of these muscle fibers. So instead of seeing it like this in the long, elongated version, you're going to see that cross-section. So this would be the circular layer. Just imagine a layer all across right here. Then you would have the longitudinal layer, like this one is oriented right down here, where you see more of the elongated fusiform shape. So when you're looking at your sample of smooth muscle tissue, a good way to tell that you're first of all in the small intestine and second of all that you're actually looking at smooth muscle is the lack of striations and the two opposing layers of smooth muscle. One in the circular layer and one in that longitudinal layer.